This is Module A8, Objective 4, and in this objective we're going to look at color as well as depth perception. In terms of light and dark adaptation, we see that the sensitivity of our visual system varies with the intensity of light. For instance, after 30 minutes in the dark, almost all of our visual pigments that were disrupted, all the rhodopsin molecules that were broken down into retinol and opsin, they have all recovered from the bleaching process, and they're fully receptive once again. This is the dark adapted state. When dark adapted, the visual system is extremely sensitive. Okay? When the lights come on from the dark adapted state, and I see this all the time in class as I turn the lights off to view the monitor. Once the light comes on, at first it almost appears as if the light is unbearably bright. Everybody is shading their eyes and squinting, trying to adjust. But again, over a few minutes of time, the sensitivity decreases as bleaching occurs. So as light hits your photopigments, we have bleaching, and that ultimately is going to decrease the sensitivity. Eventually, the rate of bleaching is balanced by the rate at which the visual pigments are being reformed. And this condition is the light adapted state. When we look at the visual pathways, we know that they began in the retina at the photoreceptors, and the pathway is going to continue to the cerebral hemispheres back in the occipital lobes. If we were to follow the pathway, we know that from the retina we have the optic nerve. I do not have my pen set up. We have the optic nerve, and those fibers will somewhat cross at the optic chiasm, and they're going to continue via the optic tract. If you actually follow the fibers that leave the eye, and I'm going to do so with just one of these, over here we have the right eye, and the fibers that leave, we see that some of the fibers will cross at the optic chiasm and head towards the opposite side of the brain. And we have other fibers that do not cross, but they actually return to the same side of the brain that the eye is located on. But either way, they're heading towards the cerebral hemispheres. When we look at cone distribution, we note that there are three different types of cones. Each cone is sensitive to a particular wavelength of light. We find the three types of cones are going to be blue cones, red cones, and green cones. And you might ask the question, well, how do we see different colors within the spectrum if we only have blue, red, and green cones? Well, the answer is the pattern and frequency of receptor activity. Again, we see this time and time again. It's the pattern and frequency of receptor activity. The textbook will note that colors like yellow are perceived through the stimulation of red and green. So it's a combination, it's a pattern, it's a frequency. Color blindness is a condition in which one or more classes of cones are non-functional. The most common type of color blindness in the population is a red-green color blindness. And in this particular type of color blindness, red lights cannot be distinguished from green light. Of course, that makes it very difficult for those who are driving. We find that 10% of males exhibit some type of color blindness, and you will learn why at the end of the second semester when we look at genetics, and particularly sex-linked genetics. Excuse me for 1 minute. There it is. Visual data. We find that visual data is combined from the two fields fields from each eye. If you were to look at, for instance, the right eye again, we see that this is the field of the right eye. While the left eye has its own field. And these particular fields, they overlap. They overlap in this region right here. So the combined fields of vision arrive at the visual cortex on the opposite occipital lobe. So what that's referring to is the left field for each eye, here and here. They both end up on the right visual cortex, while the right fields of both eye, right and right, they both end up on the left visual cortex within the brain. And it's through these fields that the brain ultimately interprets images. This is also the fact that these fields overlap here also explains why we don't exhibit a blind spot. Remember that the blind spot is caused by a lack of photoreceptors at the optic disc, and this is the site where the optic nerve penetrates the eyeball. And you have a physical blind spot, but because the blind spot for the right eye is different from the blind spot of the left eye in terms of their visual fields, we find that the left eye accommodates the blind spot of the right, and the right eye accommodates the blind spot of the left, since there is an overlap in the actual visual fields. In terms of depth perception, this is the ability to interpret three-dimensionality. Whenever we look at objects, we can note that these objects are three-dimensional. You can see that a chair is three-dimensional. I'm just using examples as I look around my office. But it's the ability to interpret that three-dimensionality among the objects within the view. And this is done by comparing the positions, the relative positions of objects between left eye and right eye images. So your brain is able to take the visual fields and interpret the three-dimensionality of the objects that you're looking at. And that ends Module A8, Objective 5, and the rest of the objectives should have been covered in the lecture.